Hey guys, so this is part 2 in the gene to protein series. It's only 2, so I don't really know if that's a series. But anyway, and this video is going to talk about translation. So translation is what happens when we go from talking in nucleic acid language to talking in amino acid language, where we completely change our premise. So we go from nucleotide stuff and nucleic acid stuff in the form of DNA and then eventually to RNA. And now we're going from RNA to amino acids. And then we're going to link those amino acids together to form a protein. So messenger RNA carries the code for proteins. It carries the information. It was made from DNA. We went through a series of changes, moving things around and protecting both ends until now we have mRNA that can now leave the nucleus and get to the ribosomes where proteins are actually going to be made. So we're going to go from speaking in our triple letters, in our codons, in those nitrogen bases, to speaking in actual amino acids like methionine and valine and glutamic acid and cytosine and stuff like that. Now there are 20 amino acids and we only have 4 nucleotide bases, A, U, C, and G. So how are we going to code for 20 different things when we only have 4 letters? Well, it's the power of mathematics, people. Lots of different ways to pretty much code for the same thing. So a messenger RNA codes for proteins based on the number of letters it can read at a time. So it can read in triplets, also called a codon, which is just a series of 3 nitrogen bases. So like if you look at this part of the picture right here, this is our messenger RNA, and this group of 3 letters refers to what we call a codon or a triplet. So here we have 1, 2, 3, 4, 5, 6, 7 codons. And those codons all code for a particular amino acid, methionine, arginine, valine, etc., etc. When you put those together, you actually make or build your protein. Now, this diagram right here should look familiar to you. It is our amino acid chart. Now, there are 20 amino acids, but there's 64 possible codes, which means that some codes actually code for the same amino acid more than 1 or even 2 times. All life is based on this particular code. It's based on these same letters and based on these same amino acids. So this is one of the strongest pieces of evidence or pieces of support that we have for a common origin for all life, because it doesn't matter if you're an amoeba or a fennec fox or a blue whale, you still have proline and leucine and A's and T's and C's and G's and U's. Now, what does this tell us? This tells us that the code is redundant. There are several codons for each particular amino acid, with maybe the exception of our start codon. There's one codon for start, which is methionine or AGU. But everyone else has several. If you look at arginine, there are 4 different ways we can make, actually there's 6 different ways we can make arginine. 4 here and 2 there. Here are all the ways we can make glycine, all the ways we can make alanine. So there's several ways we can put those groups of 3 letters together to end up with the same amino acid ultimately. We do have a start codon, which like I said before is methionine, also known as AUG, that's the code for it. And we have 3 stop codons. They don't have a special name other than just stop. There is UGA, UAA, and UAG. They all code for stop, meaning stop making this protein now. So how do we match up the amino acids with the codes that code for them? Well, through another type of RNA. This time, this RNA is called transfer RNA. So transfer RNA has the ability to read the codons on messenger RNA and pair them up or complement them with what we call an anticodon, a complementary codon. It's another group of 3 letters. It's complementary to the codon on messenger RNA. Attached to the anticodon is the amino acid that this messenger RNA codes for. So that's how the amino acids get brought in. Excuse me. So let's look at tRNA a little closer. It has a very unique structure. It is still single-stranded, but because RNA has the ability to fold and base pair with different sections of itself, transfer RNA takes on this twisted shape, which we normally describe as being a clover leaf structure. The anticodon is found on the head of the tRNA molecule. So down here in this green region, that would be the head. This is the actual molecule. This is the cartoon representation, this and this. And the amino acid is attached on the 3' end of the tRNA. So this tail region right here is where the amino acid that this corresponds to goes. So you have the anticodon on one end and the amino acid that it codes for on the other. So they all come in together as one unit. Sorry, I'm dropping stuff. So how does it work? Well, of course, there is an enzyme. This enzyme is called aminoacyl tRNA synthetase. And all that it does is it bonds the amino acid that's supposed to go with the anticodon to tRNA. Now, the bond does require energy, so we're going to be breaking down some ATP, but the resulting bond between the tRNA and the amino acid are very unstable, so it's easy for the amino acid to release to be released from the tRNA once it gets to its proper location within that chain. Okay, so you would have learned at some point that ribosomes are the site of protein synthesis, and it's true. I always think of the ribosome as being just the factory at which protein synthesis takes place. It doesn't actually make or allow for protein synthesis to happen. Like the ribosome isn't controlling any aspect of protein synthesis more than it's giving it a location for protein synthesis to happen. So it is the coupling, it allows for the coupling of tRNA's anticodon to the mRNA codon. So this has left a big debate where some biologists don't agree that a ribosome is a organelle because it doesn't actually perform a actual function. It's the site of a function, but the thing itself, the ribosome itself, isn't performing the function. So some people describe it as an enzyme. I prefer to describe it as a structure, but that's just me. So in terms of its actual structure and what it looks like, ribosomes are made up of ribosomal RNA, also known as rRNA, and proteins. And it comes in two pieces, what we call a large subunit, or the head region, the big region on top, and a small subunit, the smaller region on the bottom. And this is how they fit together. So this would be the large subunit, down here would be the small, and when you put them together, that's what they look like. So inside of the ribosome, there are actually 3 sections or 3 sites, and at each site something different is going to happen in the process of building this protein. So the first site is called the A site, and it stands for the aminoacyl tRNA site, and it's the area of the ribosome that is going to hold the tRNA carrying the next amino acid that's about to be added to the chain. So in the A site, the amino acid that we're about to add, that's where it's going to sit until it's needed. The next site is called the P site, so it's just up from the A site, and that stands for peptidal tRNA site. The P site holds the tRNA that's carrying the growing polypeptide chain. So the tRNA that's holding on to this chain of amino acids that are being pushed together is held in the P site. Next is the E site, or the exit site. The name says it all. This is where the tRNA that has already given up its amino acid is going to move into so that it can actually leave or exit the ribosome and go collect another amino acid and then come back. So A, P, and E. A site, we're sitting next in line. We're waiting to add our amino acid in. P site, we are holding on to the entire chain of amino acids, also known as a polypeptide. E site, we've already attached our amino acid to the growing polypeptide chain, and now we're just waiting to leave so that we can go continue our work. So how do we build this polypeptide? Well, it happens in 3 steps, almost like everything in biology. The first step is called the initiation step. And in the initiation step, that's where everything is going to come together. So the mRNA is going to get sandwiched in between the large subunit and the small subunit. And then our very first, or our initiator, tRNA, is going to start off the process. So here is the large subunit. This is the small subunit, and this pink strip represents our mRNA. And like I said, it's sandwiched in between the large and small subunit. That's initiation. The second step is elongation, and elongation is just adding the amino acids based on the sequence of codons that we read using mRNA as our guide. The last step is termination. When we get to the code or the codon that codes for stop, production stops because we've made everything that we need. So now that we've made this protein, well, what happens to it? Well, proteins are made to be used, so the protein needs to be sent wherever it is it needs to go. If it's going to be secreted, if it's going back to the nucleus or to the mitochondria or to the chloroplast, to the membrane, to the cytoplasm, or even outside to a different kind of cell altogether. Well, in order for the cell to know what the next step is, we need some kind of indication. So I like to think of it as I'm sending a letter to my mom in Grenada. I know that I want it to go to Grenada. I know that I want it to go to my mother. But I'm not the one who's really going to be sending this letter. I just kind of put it, in the post office or in my post box or whatever. If the people at the post office can't tell where this letter needs to go, the letter doesn't go anywhere. So it's kind of the same thing with this protein. There needs to be a little indicator that tells the parts and pieces this protein is going to this location. So just like my address label would tell the people in the post office that the letter is going to my mom in Grenada, a signal peptide is attached to the protein that we've just made, and it is almost like the address label for that protein. It tells the rest of the cell, the parts and pieces working on moving these proteins around, where this particular protein is going to go. It's its little address label. Send me to the nucleus. Send me to a liver cell. Send me to the mitochondria. So that's the entire thing, the entire story, starting with transcription all the way through translation. So let's see if we can go through it. Protein synthesis is 2 parts, transcription and translation. Transcription starts in the nucleus with DNA. DNA opens up a section. The section that codes for the particular gene that we want is selected. That is known as the template strand. The strand that we don't reproduce or we don't code is known as the coding strand. And the enzyme RNA polymerase, along with all of its transcription factors, are going to bind at a promoter site. Specifically, they're looking for the TATA box. And they're going to also bind at the enhancer, which is going to make the process more efficient. And transcription takes place, where RNA polymerase brings in complementary RNA bases for the piece of DNA that we've exposed. The first piece of RNA that we make is going to be what we call our primary transcript. It contains both introns and exons, exons being the coding pieces of genetic material, whereas introns are the pieces we don't necessarily want. They don't code for anything for this particular gene, or protein, sorry. So the introns need to be removed, and they're done, or this is done, by SNRPs, which are a combination of small nuclear RNAs and proteins. They make incisions on either side of the introns and they push them together, thereby cutting the intron out and pushing the exons closer together so that we have one long piece of exon without all of these interruptions. Now, the messenger RNA needs to leave the nucleus and go into the cytoplasm to get to its ribosome. There are enzymes in the cytoplasm that could potentially destroy our piece of mRNA, so we have to protect it. So along with removing the introns, the other two things that we will do after transcription has taken place is we will fit a GTP cap on the 5' end of the mRNA, and then we will fit a poly A tail on the 3' end of the RNA. So now that it's ready to travel through the cytoplasm, it does so until it gets to a ribosome, and it's going to get sandwiched in between the large and small subunits of the ribosomes, and then it is going to be read pretty much so that we can build a protein or polypeptide chain based on the codons of mRNA. So tRNAs get fitted with their particular amino acids by the enzyme aminoacyl tRNA synthase, and they travel to the large subunit of the ribosome. They enter at the A site, where they are waiting to add their amino acid to the growing polypeptide chain. They then move on to the P site, where they've now added their amino acid. Once they've done that, they move on to the E site, where they can then exit the ribosome, and go back and pick up another amino acid and come back in. And this keeps going until we get to an end codon or a stop codon, at which point we're going to stop. We're going to label our polypeptide with a signal peptide, and it will be sent off to whatever part of the cell or even out of the cell to do the job it was intended to do. And that is the process of protein synthesis. Alright, so this picture is just to kind of show you the entire thing in terms of a gene. So here we have our enhancer region, which is above the actual gene. Here's the ta-ta box where our RNA polymerase is going to come in with its transcription factors. Remember, it's like choosing restaurants in the Caribbean. You look for the ones that have lots of people, and that's the one that you go to. So RNA polymerase is going to look for where all of those transcription fibers line up, and that's where it's going to bind. And this region right here is the actual start. Excuse me. So we're going to go ahead and transcribe the entire thing, but we will transcribe exons and introns all at the same time. The introns will be spliced out, leaving us with just the exons. We're going to go ahead and add 3' cap, poly A tail. This whole piece that actually has worthwhile information is going to be transcribed at the ribosome, and then we'll have a protein. So this is how we go from initial piece of DNA to pre-mRNA to mature RNA that's ready to leave the nucleus. Okay, now let's talk a little bit about how this happens in prokaryotes, because there is a difference. Prokaryotes have no nucleus, so we're going to have to do some things that are slightly different here. So when we look at gene expression or protein synthesis in prokaryotes versus eukaryotes, we have to remember that in prokaryotes, the DNA is in the cytoplasm, and it's circular, and it's naked. Naked meaning it has no histones. It also doesn't have any introns. Everything is important. And if you think of prokaryotic DNA, it's a lot smaller than eukaryotic DNA. So versus eukaryotic DNA, which is in the nucleus, is linear and is arranged into chromosomes, and is wound around histone proteins. And this DNA does have introns and exons. So right away we can see that there are going to be some differences. Now in prokaryotes, transcription and translation are simultaneous. And here's an example of how it happens in bacteria. So the DNA is in the cytoplasm. We don't need to edit the mRNA because there's no introns. There's nothing to cut out. And because the DNA has always existed in the cytoplasm, there are also no enzymes that will chop it up, because if there were, the DNA would have been chopped up already. Now, Once we've translated the section that codes for our particular gene, then ribosomes are just going to read that mRNA as it's being transcribed. Like it's literally being transcribed and fed straight into a ribosome. And we're going to make our polypeptide pretty much the same way that it gets made in eukaryotes. It just happens straight from transcription, straight into translation. As parts of it are still being transcribed, the first parts that were made are already being translated. Okay, so what does this mean in terms of time? Well, there is a time factor and a physical separation factor that you have to worry about in eukaryotes that we don't have to in prokaryotes, because the DNA is not separated from the ribosomes. It's all in the same area. Eukaryotic DNA takes about an hour to go from DNA to a protein, whereas in prokaryotes, it's a lot shorter because you don't have to move the messenger RNA around as much, and there is no RNA processing because it's not needed. All right, I hope this was fairly clear and easy to understand. Let me know if there are any questions. See you guys soon. Bye.